So children, today's video we are going to study a new topic, okay, that is DNA fingerprinting. Okay, so what do you mean by fingerprinting? Okay, sometimes you would have heard from crime scenes and all, okay, fingerprints of means fingerprints police used to collect, isn't it? So what for it is done? Later, if any suspects are there, okay, it can be matched with the fingerprints that we get from the crime scenes, isn't it? So in the same way, so there are certain limitations for this. What are the limitations? Okay, it may get wiped by anyone, okay, then or else it may if the culprit undergo some plastic surgery, how they will get the real or that time it cannot be successful, isn't it? So in that cases, okay, a better type of technique is there. Now you would have heard about DNA test. Okay, so DNA test means what? DNA fingerprinting is done. So see, this is more reliable technique. Why? The DNA of a person will be there in all the cells, isn't it? It can be found from a small sample of tissues or cells, okay, like what it can be obtained from the hair follicles, skin, bones, saliva, sperms, isn't it, or blood. So in the crime scenes they used to leave this kind of signs, or some cells might be there, some blood stains will be there. So from this DNA can be recovered, okay, and it can be used for identifying the culprit later. So this is one use of DNA fingerprinting, that is in forensic science. Another important field where DNA fingerprinting is used is what in the case of paternity disputes. Means what? The parent, especially father, okay, if the father is not accepting the paternity of a particular child, okay, in that time they can use what? This DNA fingerprinting. Okay, so here DNA fingerprinting is based on certain facts, that is 99.9% of DNA of two individuals are exactly similar, okay, of human being. Two individuals they will have exact, means in a species if you take the individuals, DNA will be almost same, okay. So in human beings it is 99.9% of base sequences of human beings is same. Then how the difference will be, where this DNA fingerprinting can be used? So I told 99.9% is same. So 0.1, okay, difference is there between two different individuals, and that differences in sequence of DNA make every individual unique, okay, in their phenotypic appearance. Is it clear? So because of that 0.1 clear percent of base sequences differ from each other, each person is what? Unique. Is it clear? So if anyone aim to find the genetic difference between two individuals by sequencing the whole DNA, so we have studied what is the length of DNA, and human genome project we have studied, isn't it? It took so much years to complete, it was so costly, so it is not practicably applicable to what? To find or to compare the whole two sets of DNA, as it contains 3.3 into 10 raised to the power 9 base pairs. That much long DNA it is not possible to sequence and compare every time. Is it clear? So DNA fingerprinting identifies or focus on certain specific regions in DNA that are known as repetitive DNA. Okay, so that is depending, this technique entirely is depending on a what kind of fragments known as repetitive DNA. Is it clear? Then, so what do you mean by repetitive DNA? A repetitive DNA is some sequences of DNA that repeats many times. Understood? It's small stretch of DNA that repeats many times are called what? Repetitive DNA. For example, 80 likewise many times, or CG or ATC likewise. Clear? Or some specific sequence will be repeated many times. That kind of part of DNA is known as what? Repetitive DNA. Okay, so repetitive DNA can be separated from the bulk genomic DNA by a process called density gradient centrifugation. Density gradient centrifugation. So when centrifugation, the material will be rotated at very high speed. So when they do likewise, the bulk DNA, that is the normal DNA, will get separated from the repetitive DNA, and they form two peaks. That is, the bulk DNA form the major peak, and the small the other DNA, that repetitive DNA, form the small peaks. And that small peaks are known as satellite DNA, which contain what? The repetitive type of DNA. So they are known as what? Satellite DNA. And the satellite DNA are containing what? Repetitive sequences. For example, AT reach or CG reach. So depends upon the base composition. What is the base composition in the AT reach or CG reach? Then length of the segment, how much length is there, which is the repeat, which forms the repeating unit, and the number of repetitive and the number of repetitive units. How many times it repeats. For example, sometimes it may be found 50 times, or it may be 500 times also. Simply they repeat. So that kind of DNA is known as what? Satellite DNA, and they are classified on the basis of what all? Base composition, then what? Length of the segment. Clear? And number of repetitive units. So on all this basis, they are classified into different categories, like microsatellites, mini satellites, etc. Mini satellites, etc. Is it clear? So macrosatellites means, what is it? Microsatellites means the microsatellites, they are having around 15 to 50 base pairs repeated thousands of times. Is it clear? Mini satellites. Then the microsatellites, they are having less than 10. Base pairs will be less than 10 and repeated 10 to 100 times. So that is the difference between microsatellites and mini satellites. So what is this mini satellites and microsatellites? They are satellite DNA. What is satellite DNA? The DNA which is having repeating units. Repeating base pairs. Is it clear? And this repeating base pairs, they form the minor peak in density gradient centrifugation, that are known as what? Satellite DNA. And there are of two types, depends upon what their base pairs and length, then what their repeating times, how many times it repeats, on that basis. Is it clear? Then this satellite DNA, so they are having some common features. So let's see what are the common features of satellite DNA. This was asked once. The sequences, they will never code for any proteins, and they form a large portion of human genome, and they show high degree of polymorphism and form the basis of DNA fingerprinting. So these are the characters of what? Satellite DNA. means they do not code for any proteins. I told many proteins are not coding, they are simply there. Okay, sorry, many base sequences are simply there in our genome. They are not coding for any protein. Simply they form the major portion of human genome, and what they are they shows high degree of polymorphism. So what do you mean by polymorphism? Difference. Clear? So see, the difference between the individuals, okay, is due to the difference in the genes, isn't it, or in the DNA base sequences. So especially the satellite DNA shows high degree of polymorphism. Is it clear? So this forms the basis of DNA fingerprinting. Means instead of comparing the whole set of DNA every time, the scientist or those who are doing will be focusing on what? DNA fingerprinting, those who are doing will be focusing on what? Polymorphism. So polymorphism is mainly shown, or what it can be easily studied in where satellite DNA. Is it clear? Okay. So let's study more about the DNA polymorphism. Okay, so what do you mean by DNA polymorphism? DNA polymorphism means variation at the genetic level. Okay, and this what differences arises due to what? Mutations. And this DNA polymorphism forms the basis of genetic engineering, and okay, means a basis of genetic engineering of human genome, and also it is useful in what? DNA fingerprinting. Is it clear? So I told this polymorphism occurs due to what? Mutations, isn't it? So mutations can occur in both somatic cells and in germ cells, isn't it? So as the mutation happens in the somatic cells, it will not make any difference. It cannot be inherited. So if any mutation happens, especially in the satellite DNA, okay, that will not cause any trouble to the individual, isn't it? If it is causing any trouble to the individual, it will be harmful. Otherwise, what happen? It will not be any causing any harm to the individual. Why it is not causing any harm to the individual? It is what? Repetitive DNA. It is not coding for any protein. So mutation occurs. Okay, so differences also causes. Okay, but that will not cause any trouble to the individual. So it will be what? Transmitted from parents to offspring. It can be easily transmitted from parents to offspring. And one more thing is there, as you know, this one new individual is formed from a zygotic cell. Okay, the whole cell of human being will have the same repetitive DNA. Okay, they will have the same type, so form the repetitive DNA. Okay, then so as I told, if germ cells, okay, mutation does not seriously impair individual's ability to have offspring, then it will be transferred to the next generation. And I told what the difference will be same in all the cells of a particular human being. That's what we are keen in, what? Collecting the samples from the crime scene. Okay, so it can be blood stain, it can be what? Hair follicles. Okay, so this contains cells, and the cells will have DNA of that particular person, same person. So if the same person has visited or done the crime, there is no difference in them, and this will be very, very unique. Okay, the polymorphism or that particular DNA of that particular individual will be unique. That cannot be seen in any other individual. Okay, and this polymorphism can be can be in the allelic sequence variation also, isn't it? So in allelic, if allele, do you remember what is that? Different forms of the same gene. Okay, so if the alleles are different, okay, then also it is known as what? The DNA polymorphism. Okay, so if DNA polymer, if more than one variant of a allele at a locus occurs in a human population with a frequency greater than 0.01, okay, then it can be what? It can be described as DNA polymer. Means if different alleles are occurring, okay, with a frequency greater than 0.01, okay. So this inheritable mutation, if it is available in a population with high frequency, it is defined as the DNA polymorphism, okay. So in alleles, if there is difference, okay, and in a population you can find them at a high frequency, then it is also known as what? DNA polymorphism, okay. It is known as DNA polymorphism, okay. Then I told what the probability of variation to occur in the non-coding sequences, that will be more. Why? Because if it is happening in the coding sequences, what can happen? It can impair the individual, isn't it? It can cause certain harm, so it will not be promoted to the next generation. So if this variation happens in the non-coding sequence, no harm will be there and it can be easily inherited, okay. And this mutations will keep on accumulating generation after generation, and there will be much variability or polymorphism, okay. So this polymorphism can be a single nucleotide change, okay, or it can be very large scale changes also, okay. Sometimes only in one at a time, in one sequence only the mutation can happen, but sometimes what happen, it can be a large scale, okay. Means many nucleotides can be changed. And for evolution and speciation, such polymorphism is very, very important, okay, because this accumulation of variation results in what? Evolution, isn't it? So for evolution point of view, this polymorphism is very important. Clear? And in DNA fingerprinting, okay, it is mainly depending on what? This DNA polymorphism. Because why? In certain regions only polymorphism is shown, I told, because 99.9% of DNA of two individuals are same. So the scientists, they focus on certain kind of differences, okay, that is occurring in the satellite DNA and are used. Is it clear? So the technique we will study in the next class, okay.